Hello everyone, I am Nandini M. Kager, branch Computer Science and Engineering, 3rd SEM, subject Mathematics, topic Analysis of a completely randomized design. Contents: Introduction, Completely randomized design, Advantages of a CRD, Disadvantages of a CRD, Uses of a CRD, Statistical analysis of a CRD, Anova table for a CRD, Conclusion, Reference. Introduction. Designing an experiment means deciding how the observations or measurements should be taken to answer a particular question in a valid, efficient, and economical way. A well-designed experiment helps the workers to properly partition the variation of the data into perspective component in order to draw valid conclusion. Completely randomized design. The completely randomized design is the most simplest of all the design based on a randomization and replication. In CRT, all treatments are randomly allocated among all experimental subjects. This allows every experimental unit, plot animal, soil sample, etc. to have an equal probability of receiving a treatment. Randomization procedure. Treatments are assigned to experimental units completely at a random. Every experimental unit has the same probability of receiving any treatments. Any difference among the experimental units receiving the same treatment is considered experimental error. Randomization is performed using a random number table, computer, program, etc. The advantages of a CRD. CRD has the several advantages. It is easy to lay out the design. There is a completely flexibility in the number of the treatment and number of a replication which may vary from treatment to treatment. The number of replication need to be same for each treatment. The CRD provides a maximized difference for the experiment of a experimental error. And disadvantages of a CRD. The main objection against the CRD is that the principle of local control has not been used in this design, so that the experimental error is inflected by the present of the entire variation among the experimental units. Uses of a CRD. Under a condition where the experimental material is homogeneous in a physics, chemistry, in a chemical, and in a biological experiment, in a some greenhouse studies. The next one is in a small experiment where there is a small number of a difference. CRD is a may used by in a chemical or baking experiment where the experimental units are the part of the thoroughly mixed chemical or a powder. And this is the Anova table for a CRD. And next one is assumption underlying Anova. Experimental errors are random, independently and normally distributed about a mean of a 0 and with a common variance. The above assumption can be expressed as a NID. Departure from this assumption can affect for the level of significance and the sensitivity of a F or T test to real departments from HO. This results in the rejection of HO when it is a true type of I error more often than a cells for. The next one is the power of the test also is reduced if the assumption of a NID is violated. Violation of the assumption NID with the fixed model is usually of the consequence because ANOVA is a very robust technique. Violence of the basic assumption of ANOVA can be investigated by object plots of the residuals. And the next one is conclusion. A completely randomized design relies on a randomization to control for the effect of extraneous variables. CRDs are for the studying the effect on the primary factor without the need to take other nuisance variables into account.